Assalamualaikum warahmatullahi wabarakatuh to Dr. Farah and Prof. Shahir. My name is Nurul Syafiqah binti Muhammad Ali. Today, I will talk about my proposal oral defense for the topic Microbial Diversity Analysis of Different Types of Soil Treatment. My supervisor's name is Dr. Husalina binti Husin. This is the overall overview of my slides. Introduction. Malaysia comes second in the world of oil palm plantation. In 2010, roughly 5.2 megahectare or 60% of the country's total land area were used to establish oil palm plantation. Soil fertilization in such oil palm plantations is typically conducted to boost production through increasing oil yield. This study sought to establish the relationship between the soil physical and chemical properties on microbial composition in the oil palm plantations. In particular, it focused on the soil fertilized with chemical in a long term to natural secondary soil. The Illumina method sequencing has been used for a very fast and more visible soil bacterial population and profile. Moving next is problem statement. Farmers in Malaysia use inorganic fertilizers in oil palm plantations to increase the productivity in acidic soils, which are not ideal for high yields. These fertilizers, rich in nitrogen, phosphorus, and potassium, can enhance the soil nutrients. However, long-term effects of fertilization can have negative impacts such as alters the pH of soil, reduce organic matter, and also alters the population of microbes in the soil. The use of inorganic fertilizers may lead to high production costs and environmental population. Despite these drawbacks, this study aims to measure the impact of constant use of inorganic fertilizers towards the microbial diversity in the soil. There are two objectives that we will be looked in this study. First objective is to determine how the application of inorganic fertilizers in a 25 years old oil palm plantation in Peninsular Malaysia indirectly influences the characteristics of soil and also the microbial diversity, especially of secondary forest soil. Second objective is to measure the impact of constant use of inorganic fertilizer on the physiochemical properties and microbial community structure and function of soil. In scope of study, first, sample size. Sampling involved 4 plots in the plantation and secondary forest with soil collected from two depths, 0 to 50 cm and 15 to 30 cm. Duration of study, the sampling will be conducted on the first day of study, and that is after the last fertilizer schedule has lasted so that it does not influence the soil parameters, pH, total nitrogen, total phosphorus, total organic carbon, cation, exchange capacity, and exchangeable cations such as K+, Mg2+, Ca2+. Limitation, possible contamination of the soil samples during collection or the laboratory work. Significance of study. We want to provide some important information concerning the relationship between soil physiochemical properties and the diversity of microorganisms in oil palm plantations. This study beneficial to the public by the fact that they will be enlightened on the environmental impacts of such agricultural practices. Information on data collected from such strategies can be useful in planning as well as formulating strategies to promote sustainability of land and soil. Next is the literature review. Microbial diversity. Some aspects covered in this microbial diversity include genetic diversity, which involves first, variability and distribution of genetic information. Second, species diversity, which is the variation in types of bacteria and fungi. Third, ecological diversity, which is variation in the structure, interactions, and roles of microorganisms in oil palm plantations. A parameter is inorganic fertilizer, which is fertilizers that are made from non-living materials, contain elements such as nitrogen, phosphorus, and potassium, which are essential in plant growth. Overall, we want to know if constant use of inorganic fertilizer can impact on the microbial community of soil. This is the research flowchart for the methodology. First step is experimental design and soil sampling. Surface soil samples for analysis were taken from the 0 to 50 cm and 15 to 30 cm of the soil layer of the oil palm plantation and secondary growth forest. Second is soil properties and nutrient analysis. The measurement of pH using pH meter. Total nitrogen and phosphorus contents determined using a block digester and auto-analyze. Third is polymerase chain reaction (PCR) and denaturing gradient gel electrophoresis. Amplification of the DNA sample through the polymerase chain reaction (PCR) technique with target gene-specific primers. This is the overall step for the objective 1. Moving next is fourth step is high throughput 16S rRNA gene sequencing. The samples were sequenced from the MISET sequencing platform. Lastly is statistical analysis. Analysis of variants ANOVA with the aid of the SAS software for soil properties and nutrient depletion. These two steps is required for the objective 2. This is the list of materials that will be used during conducting the experiment. There are two expected results that we will look under two objectives. First, the result for objective 1, long-term application of inorganic fertilizers resulted in significant changes in soil pH, organic carbon, and when compared to the secondary forest, total nitrogen and organic carbon decreased with inorganic fertilizer application. For the second results of objective 2, in the second growth forest, the relative abundances of acidobacteria and proteobacteria and actinobacteria were greater while the firmicutes were less in the soil after 25 years of inorganic fertilizer application. Firmicutes, bacteroidetes, chlorophyllaceae were identified as the most dominant phyla that existed in the secondary forest ground. This is the Gantt chart of FYP from October 2024 to June 2025. Here are the list of references for the slides. Thank you for listening.